Hello, everyone. Today I'm starting with a very short lecture, which I call Introduction. It's really some information that I really hope you know already or are familiar with already. This course, Bio 240, doesn't have a prerequisite in the biology department. So some of you may have taken Bio 120 already. That's perfect. Some of you may have taken Bio 230 already, but most Grossmont students take Bio 240 first. So I'm not assuming that you know this information inside and out, but you should generally be familiar with these vocabulary words and these concepts. If you have never had a biology class before, it's still possible to be successful in this class, but you'll have to do more work, right? So this little introductory lecture is to just remind you about some things in biology, hopefully. And these are basic ideas that we are going to be using during the whole semester. So if you aren't as familiar with them as you would like to be, you can go to your OpenStax textbook and you can sort of flip through it, look at things in chapters 1 through 8 and review or just make sure that you are familiar with these concepts before the semester really gets rolling. You won't be directly tested on this content, okay, but you're going to need to understand it in order to understand things that we are doing as we go through the semester. All right, so here we have the Earth, and this biology course basically deals with everything from the sun and our atmosphere all the way down to single individual organisms. So when you're going to go inside the cell and look at things of how processes inside the cell work and more about how cells talk to one another, that's going to be in your Bio 230 class. But we have a lot to cover in this class. The world is big. There's a lot in it. And so we have three main subjects. We have ecology, we have evolution, and we have organismal biology, which is not only to understand the diversity of living things, but also to understand how they go about doing the tasks of life. So we are going to look at the structures that they have, and we're going to look at the functions that those structures have and how different organisms get those jobs done. So we'll see that a snail has very different structures in its body than a human being does, but they still are going to achieve the life functions that are needed in order to do those things. So let me go back to this slide. One of the things that ecology is all about is looking for patterns. And we can see on this single slide that there's some different colors on the image. And if you know almost nothing about biology, you might guess from where those patches of green are that those are tropical areas with lots of rainfall. They might have a lot more plants. They might be doing more photosynthesis. So why do we have more photosynthesis happening in those areas compared to others? That's the kind of question that we're interested in talking about in ecology. As far as evolution goes, we will come back to that in a lot more detail. That's the first unit that we're going to talk about. So hopefully you have a little bit of an idea what I mean when I say that already. We want to start by defining life. When we're studying biology, we're studying living things. So what do all living things have in common? The first characteristic is that they are made of a cell, right? So we have to define what a cell is, and there's a sample cell on this diagram. So if you look at it carefully, think for a minute whether you think that is a prokaryotic cell or a eukaryotic cell. All right, that's right. It's a prokaryotic cell. It doesn't have a special section that's partitioned off with membrane to make up the nucleus. That's one of the hallmarks of a eukaryotic cell. But both prokaryotic and eukaryotic cells have three basic components. They might have other things too, but we are going to find that all cells are going to have a plasma membrane. They are going to have ribosomes, and they are going to have DNA. The space inside the cell is filled up with a liquidy stuff called cytoplasm. And I find that in these lists of what cells have, the cytoplasm kind of goes without saying. But in this diagram, what's with all the empty space represent? It's the liquid environment inside of the cell. Now, the inside of that cell is mostly water. So we'll be talking about solutions that are based on water as the environment of life. So not only does the outside of living cells need to be either water, or that cell has to have a protective waterproof covering, and the insides of cells are always going to have a watery solution that we call cytoplasm in them. All right, so both prokaryotic and eukaryotic cells are going to have a plasma membrane or cell membrane. They're going to have information stored in the DNA molecule, and they're going to have these structures called ribosomes. So if you have had Bio 230 or you remember it from Bio 240, the reason why all cells have ribosomes is because of the job that they have. So what is their function? Well, they're going to take the message that's stored in the DNA, and once it's made into a messenger RNA, the ribosome is the place where that message is going to be made into a protein, right? So we can't use the DNA unless we have ribosomes to turn that information into proteins. So there's some other goodies here. We'll come back to those as we talk about prokaryotic and eukaryotic cells later on in the semester. Right, so we're going to define living things as having a cell, as having DNA, and also a variety of other characteristics. Different textbook authors are going to give you slightly different lists. I personally like this list because we can make a word that says charm, and charm is nice, charm is charming. And so we can remember that there is a list of 5 things that we're going to examine something and decide whether it is a living thing based on these characteristics. The first one for the C is complexity. Our understanding of what complexity is doesn't have to be complex. All that means is that the living thing is going to be organized in some way. It's going to have some kind of pattern, and we'll see that molecules are assembled into structures such as the cell membrane, and that the organism has to use energy in order to make order out of the disorder that molecules would have if they were just floating around by themselves. And so when we look across a landscape, we may see a rock, we may see a house, we may see a tree, and we can determine that both the house and the tree have complexity, right? But the house it has complexity, it has order, it has an organization, it has a plan because humans designed it, right? The tree has complexity that was built from the instructions in its DNA. The next characteristic for the H is homeostasis. If you think about what homeostasis means, stasis means staying. And so homeostasis is the ability that living things have to keep themselves, whether it's a single cell or a larger organism, within a range of characteristics that are suitable for life. So we often take an example like body temperature in humans. That is, our bodies are able to regulate our internal body temperature. So we use energy to do work in order to keep our internal temperature within a set range. It isn't exactly perfect all the time. It may go up and down a little bit, but it isn't going to change as much as the environment changes. So one of the fundamental things that all organisms have to keep regulated is the amount of water that they have inside each cell. And so homeostasis of water we often talk about as water balance or osmoregulation, right? So that's an example of a kind of homeostasis. Your body's doing homeostasis all the time. If you're not doing homeostasis, you're dead. All right, so It's an important idea that living things are able to regulate their internal conditions in a manner that's going to keep them alive, right? And if we don't do that, we're dead. The next characteristic is adaptation. And we're going to use this, the A, letter A for adaptation, in a way that is specifically related to evolutionary changes. When we start talking about evolution, we'll see that it is the DNA molecule that's going to change in some way from one generation to the next, or the proportion of individuals in the population that have some characteristic that's going to change from one generation to the next. So having DNA is kind of a way that we can make sure that our organism is a true living thing. It's going to have that capacity for adaptation. Reproduction, the word with an R for charm, is going to be essential. We aren't going to keep our species around for very long if it isn't capable of producing offspring like itself. All right, so we're also going to start right away at the beginning of the semester talking about reproduction because it ties into so many different processes that we also are going to talk about right away. So reproduction is having offspring that are like yourself. Metabolism is the M word. So metabolism is an idea that has to do with how organisms take in and use energy. So if you thought about metabolism before as, say, something that happens that has to do with mitochondria or with chloroplasts, that's a great start. In Bio 240, we'll be expanding to include not just energy metabolism, but also all the chemical reactions that go on inside of a living thing. So it isn't just energy processing, it's all the processing of food molecules into other things that you might need, removal or management of waste products. So there's really a vast array, but basically metabolism has to do with taking molecules in, transforming them in a way that's necessary to get what you need, and then you make waste products as a result of that. All right. So these are the characteristics that we're going to apply to something and decide whether it's a living thing or not. So if we look at a bunny rabbit, we can see that it's organized, that it's going to be doing homeostasis, it can adapt over time, it's going to reproduce, it's doing metabolism. Isn't as easy to observe these things in single cells, but we know that they do those things. Our rock is not going to do any of those things. It is inert, right? So it can't do chemical reactions can happen to particles, but it doesn't have any unique identity that's going to allow it to procreate itself, for example. The minerals that make up the rock may be in a particular pattern, so it may have complexity as well, right? But it doesn't have all of these characteristics. Now, a tricky group of organisms to think about whether they're alive or whether they're not alive is viruses. And we have the coronavirus running rampant through human populations right now, so it's a good time to talk a little bit about what a virus is. I'm not going to spend too much time though. I'm actually going to have you read a chapter chapter section in the textbook about this. It will be important for you in our online environment to be doing some textbook reading. And there's different ways that I'll be incorporating that into our pathway through the semester for you. The textbook, however, is free and it's available online. So I have links for you in the Canvas modules to the chapters that I'm asking you to read. So viruses are interesting because for them they're basically an inert particle. They're a protein coating floating around in the environment with a little piece of DNA or RNA inside of it. And it fastens on to a living cell. It injects the DNA or RNA into the cell. It uses the ribosomes that are inside the cell and the energy that is inside the cell. in order to make copies of itself, and it will burst out of the host cell once it's made lots of copies. So that's how it damages the cells in your body. It's basically a cellular parasite. So it has complexity because if you look at the capsules, the protein molecules are ordered. There are specific shapes for each kind of virus. We can take electron micrographs of them. We can see their structure. It can't do homeostasis. In fact, it has no chemical reactions that happen inside of it. It has no way to take in energy on its own, and it doesn't regulate its own internal conditions. It's very resistant to difficult environmental conditions, however. All right, so as far as adaptation goes, think about that for a minute. Can a virus adapt? Can it change its characteristics? And the answer to that question is not very far away in our minds when we think about the Delta variant that's spreading through our population as I speak. And the reason it's a variant is because its DNA or RNA has changed. It's had mutations. And so it has slightly different characteristics, and we can sequence its genome and we can see that it's different from other strains of the virus and that it has different characteristics as a result of that. So viruses are tricky because they have complexity and they can adapt, but they can't do homeostasis, reproduction, or metabolism by themselves. They need to have a host in order to accomplish those tasks. All right, so when we think about living things, the other thing we want to make sure we're clear on is that death is the end of life in living things, and that's when the organism is no longer able to carry on these critical life functions, right? So you don't necessarily die if you can't reproduce, but you do die if you can't do homeostasis and or metabolism for some reason. All right, if you are starving, you don't have enough food, you will eventually die, just like your cell phone dies when it runs out of battery. Or if your body is injured or damaged in some way, or an organism, the cell is injured or damaged in some way, that prevents it from doing homeostasis, that prevents it from doing one of the basic life functions. So we'll talk about that later on. Some organisms can live for a very, very long time, but there is always some way that it's possible that can end. All right, so we are going to define the smallest kind of living thing then as a single cell. It's going to be separated from the environment around it by a cell membrane. Now we're large, complex organisms with many, many cells. They are stuck together at their cell membranes. So the same processes happen inside our own bodies, but it's easy to think about this with single-celled things to start with. All right, so getting things into and out of the cell is going to be incredibly important in everything we talk about with physiology later on in the semester. So remember, the cell membrane is made of two layers of phospholipid molecules. They have the water-loving or hydrophilic phosphate heads towards the watery side. Now, when we have a cell membrane, both the outside of the cell and the inside of the cell are watery, right? They're water solutions. And then they have the hydrophobic fatty acid tails that are nonpolar pointing into the middle. So we have a two-layer cell membrane. And in order for things to get through it, these characteristics of the whether molecules are hydrophilic or hydrophobic are very important in determining how they get through the cell membrane. There are also things that are different about how very large molecules get into and out of the cell versus smaller molecules. So when we have a larger chunk of stuff, say a bundle of protein molecules that we want to export from a cell, we're going to find them inside the cell in a little ball of membrane. So a little ball of membrane is going to be called a vesicle. And if that vesicle moves to the surface of the cell membrane, because of the fluid property of the cell membrane. That means all those little phospholipid molecules can move a little bit with respect to one another, right? As it gets to the surface of the cell membrane, it's going to merge with the cell membrane and it's going to become part of the cell membrane. And once that happens, the stuff that was inside of it is going to be on the outside of it. So that process, when we're going something outside the cell, like when you go to an exit in the movie theater, that's exocytosis, stuff's going out. The opposite process is going to happen. The cell membrane basically can grab onto stuff it wants inside the cell and move it inside in the reverse process. That's called endo. So endocytosis is going to be moving things into the cell. Exocytosis is going to move things out of the cell. If you're a single-celled organism, this is how you eat and you poop. You're going to get stuff and bring it into the cell and get waste products and move them out. All right, so some molecules are going to be able to move across the cell membrane. They're very small. If they're polar, they tend to be able to move through the cell membrane. They'll kind of squirt through those phospholipid tails. So molecules that can move straight through the cell membrane are going to move through based on the process of diffusion. So they'll go down their concentration gradient from one side to the other. Molecules that either can't move through the cell membrane or that the cell wants to very carefully regulate, it can put a protein, shown in purple in this diagram, going through the cell membrane, and it basically makes a little tube. And the properties of the inside of the tube are going to be compatible with the structure of this molecule that it wants to be able to move through. So if there's this little tube that's special to this particular molecule, then it's going to be a lot easier for it to move through the cell membrane. But it's still going to go down its concentration gradient, right? So this little tube isn't pumping or moving those molecules back and forth. It's just letting them go down their concentration gradient faster and more easily. All right, so we call diffusion and facilitated diffusion, which is when there's that tube protein. We call those passive transport because no energy is being used. We're going to call it active transport if energy is being used. Energy is going to be in the form of an ATP molecule. We're just going to deliver the energy needed. And in that case, the protein, we're still going to have a protein, and it is somehow going to be able to grab hold of this molecule and push it to the other side. And it doesn't matter if there's a concentration gradient going one way or the other, it's going to pump or move these molecules through as long as there is energy available and the cell wants to be doing that. So that's going to be called active transport, and it's different because it can move molecules up their concentration gradient, but it requires energy in order to do so. All right, so we call all of these different kinds of transport transmembrane. That means going across the cell membrane. So anything that needs to go into or out of a cell is going to have to be done by one of these forms of transmembrane transport. Right, and we can also call exocytosis secretion, and we can also call endocytosis absorption. If our diffusion is happening with a water molecule, if that little black dot is representing a water molecule, it has a special name and it's called osmosis. So water is going to go down its concentration gradient just like any other molecule will. So because the cell membrane is able to regulate what crosses it, then we say the membrane is selectively permeable. It's saying, okay, I want these things to go through in this amount, and I don't want these other things to go through. All right, so permeable means things can pass through. Selectively means that the cell gets to choose what things are going into and out of. All right, so this is all of these membrane transport processes have to do with the life processes that have to do with homeostasis, because they're going to be regulating what things are going into and going out of the cell. All right. So when we look at living things, they are going to be made up of molecules. And the reason you have to take chemistry as a biology major is that living things are made up of molecules. And it turns out that they are very small molecules are built up together into bigger molecules. And then bigger molecules are often built up into even bigger molecules. And then those things are built up into other structures we start to think about as the kinds of structures that we more interested in, like organelles, right? So when we think about a heart beating, the heart is muscle tissue, and those muscle cells have some structure inside of them that is made up of protein molecules that is actually doing the work of the contractions that the whole heart is making, right? So we still have to be thinking about molecules when we're thinking about how living things are built and how they do their work. So we have 4 different kinds of biological molecules. They're called macromolecules because they're very large. And we have a series of structures that make up these kinds of molecules. So there's a single smallest version of that molecule that's a component part. We call that a monomer. Those monomers are fastened together to make a dimer if we only have 2 together. And then when we have many fastened together, we have a polymer. So the macromolecules usually refer to sort of the polymer form, and then they're going to build themselves into something that we see in an actual cell level structure. So our 4 groups are amino acids. Proteins are at the top. Proteins, which have as their monomer amino acids. When we put more than one amino acid together, we get a polypeptide. We also call that a protein. All right, so our proteins are one of our categories of macromolecules, and their subunit is the amino acid. Another kind of macromolecule are the nucleic acids. Now, nucleic acids include DNA and RNA. And so DNA means deoxyribonucleic acid. So that nucleic acid part in there means that it's made up of subunits called nucleotides. Each nucleotide is made up of 3 parts: a phosphate, a sugar, and a base. Right, so our nucleotides are built up into DNA strands, and then the DNA strands form chromosomes, which are much larger structures. Right, we can also build RNA strands out of our nucleotides as well. Our next class of biological macromolecules is the carbohydrates. And carbohydrates are also often known as sugars or starches. So the single units of our carbohydrates are usually called single sugars. An example is a monosaccharide, such as glucose. And a lot of these are 5 or 6 ring molecules. We'll be talking a lot about glucose various times during the semester. So these glucose molecules are fastened together into very, very large molecules that we are going to find in the bodies of plants, much more than in the bodies of animals. We do find glucose circulating in our bloodstream because it's a molecule that has a lot of energy stored in it. And that's the molecule that's going to go into mitochondria to get broken down to release that energy. The last group of biological macromolecules is lipids. And an example of a lipid is shown in this figure. It has a subcomponent or a subunit called a fatty acid. Often we talk about those fatty acid tails. So these are long chains of carbons, and depending on how many hydrogens they have on them, they have different properties. So we have triglycerides as an example of a lipid molecule. They're called triglycerides because they have 3 glycerols across the top, and then they have 3 fatty acid tails. And this is the way that fats are stored in our bodies. All right, so important take-home message here. You should be familiar with the idea that there's 4 different kinds of biological macromolecules and the basic properties of them. We'll be talking about them. I won't make you draw their structures as we go through the semester. All right, so the way that we're going to get the information that's stored in the DNA molecule, right? And so the DNA molecule is going to be a strand of these nucleotides, and it's going to have the letters of A, T, G, and C that are those nitrogenous bases that we find as part of the nucleotides. Those A's, T's, G's, and C's make up a message. The message, this long string of them, we're going to call it a gene, right? So a gene is going to eventually be used to make a particular protein. Each gene makes one particular protein. That protein is going to have a job to do in the cell. So the way we get the information from the DNA molecule out into the cell where we can use it is first the process called transcription. Right, so if you're transcribing your notes, you wrote them in class, they're real sloppy, you're just going to rewrite them in the same language. Right, we're going to use nucleotides to write a copy from the DNA molecule. We don't want to let the DNA molecule out of the nucleus. It's precious. It's like those rare books in the library. They don't let you check out. Right, we don't want to take it out of the nucleus. We're just going to unzip it. We're going to copy off the section we need. That's going to make a messenger RNA. That messenger RNA is going to move outside the nucleus, and it's going to go to a ribosome in order to be translated into a protein. So translation, if you think about that word going from one language to another one, in biological macromolecule language, we're going from nucleotide language, right? We were in nucleotide language when we were a DNA. We're in nucleotide language when we were an RNA. Right, but now we're changing languages. So that process of translation, going from RNA to protein, we're turning the message that's spelled out in letters signifying nucleic acids, we're turning them into amino acid message. Right, so every 3 bases in the RNA makes up a codon. Each codon codes for a particular amino acid. Right, you will talk about this until you are sick of it in your Bio 230 class, but I want you to remember the basic idea of what's happening here. We'll talk a very, very little bit about how processes in cells and in organisms are regulated based on how the proteins are produced. Right, our last topic to make sure that everybody's ready to go with Bio 240 stuff is talking about energy and chemical reactions. So chemical reactions are going to be things that happen that make or break, or do both, the chemical bonds between atoms in a molecule. Each chemical reaction is going to either require heat or energy to put into it, or it's going to release energy. All right. So the vast majority of chemical reactions that happen in the bodies of living things are going to need energy put into them. Right. So we eat food so that we have energy. Plants do photosynthesis so that they have energy that they can use to do work in the cell or in the body, right? So we're going to have 2 different kinds of energy. Remember from your physics class, we have potential energy and we have kinetic energy. Potential energy is energy that's stored. If you push on a spring, it will use that energy to do work if you let it go, but as long as you're holding the spring squished, it has potential energy in it. So the energy that's stored in chemical bonds is potential energy. Also the energy stored in batteries, for example. It's just waiting there, ready to be used. Kinetic energy is the energy of movement, and so that's going to be the energy that's actually causing something to move, and it gets used up by doing that movement. So remember that there's 2 laws of thermodynamics. I'm not too concerned with the exact phrasing of these laws, but the basic idea of the first law is that energy can't be created or destroyed. that when we have a chemical reaction, that energy is going to be transformed, right? It isn't gone, right? It just goes into a different form. So when we have a chemical reaction, we're going to, if we break a chemical bond, then the energy that was stored in it has to go somewhere. It may be released as heat, right? So that's a form of energy that's just going to warm up the environment around it. Or it can be transformed into potential energy stored in a different chemical bond. So we're looking at a chemical reaction on this slide that's a fundamentally important one for all living things, which is the process of cellular respiration. So even plants do this. They busily make lots of glucoses by doing photosynthesis, and then they have to go through the process of cellular respiration in order to get the energy out of the glucoses again. It's pretty silly, but that's what they do. All right, so we've got a glucose molecule on the left-hand side. That is a reactant. We are going to combine it together with oxygen, do a whole bunch of stuff you're going to talk about in Bio 230, and end up releasing some of the energy as heat, and then transforming the rest of the energy into potential energy stored in ATP molecules that can then go up and deliver that energy in order to do work in the cell somewhere. So energy that we think of being used to do something in our bodies, for example, going for a run, our muscles are contracting. And the reason the muscles contract is because they have a gazillion little ATPs in the muscle cells providing the energy for those muscle contractions to happen. So we have products of this chemical reaction. We've got carbon dioxide and we've got water. This is a balanced chemical reaction. So we've reorganized the carbons and the oxygens from how they started and the hydrogens, and we have transformed them into something different. Right now, our second law of thermodynamics says we don't get something for nothing. Basically, that in any chemical reaction, that some of that energy is going to be lost as heat. Now, this cellular respiration chemical reaction is extremely efficient compared to chemical reactions that are engineered by humans in order to be used to do work. For example, when you burn gasoline in your car engine in order to turn the potential energy stored in those hydrocarbon molecules, which are not dissimilar to glucose in the way they store energy. It's going to get converted into the kinetic energy that makes your wheels go around and makes your car go forward. But what happens to the engine of the car is that it heats up, right? So that's reflecting the heat that's lost from that combustion reaction, right? So those chemical reactions are going to be critical to all of those metabolic processes that are the things that have to happen in the bodies of living things or in the cells of living things in order to make that happen. So one of the things I hear Bio 240 students complaining about all the time is all the math and chemistry and physics classes that you have to take, but it's really critical to understand basic math and physics and chemistry because without them, we can't understand how living things' bodies work. So biology majors are the superheroes of STEM. Math majors just have to learn math. Physics majors just have to learn math and physics. Chemistry majors just have to learn math, chemistry, and physics. But biology students have to learn all of that. And on top of that, memorize a whole bunch of stuff. That's the biology. Okay. So as you're going through this semester, I hope you'll be making connections between what we're learning in this class and in your other science classes and how useful that is to you. And we'll talk about a lot more bigger picture things when we get going on class the rest of this semester. So hopefully you were familiar with the topics I talked about today. You're getting an idea of how I lecture, how I try to explain things so that you understand them, and try to focus on things that are important for what we're trying to learn in this class. All right, so there won't be a test worth points on this material, but we may be referring to it, or I may assume that you have a general understanding of it as we proceed through the semester.